what happens, when that cell division occurs and we get some twins here.
Into two.
Oh, double trouble.
Yeah.
Yes.
Watch out.
But if you remember last week, we talked a little bit about meiosis and how the sex cells are formed, and then they recombine to form cells. So we're going to talk about what the odds are and probabilities of different characteristics appearing at first with Mendel's crosses of plants, but then we're going to talk about people. So a little bit later, I'm going to ask you two to come back, and we are going to look at some of these double characteristics that you two share.
Okay, good.
Thank you. Okay, last week we talked a little bit about meiosis and mitosis cell division, which leads us into today's lesson, which deals with when these cells get back together to form a human being or a plant or any other animal. We're talking about sexual reproduction. And the California state standard that we are dealing with today is a standard about genetics, and it states that a typical cell of any organism contains genetic instructions that specify its traits. And it goes on to say that an inherited trait can be determined by one or more genes. And that plant and animal cells typically contain 2 copies of every gene. These copies, or alleles, may or may not be identical. One may be dominant and one may be recessive in determining the phenotype. So we're going to talk about all these standards today, and we're going to go through some activities that show us how these standards work and how to actually put them into a pictorial form so you can better understand Mendel's experiment. And so before we begin, I guess I should give you a little bit of history about who Gregor Mendel was. Gregor Mendel was an Austrian monk. He worked in a monastery. And in 1851, he went to the University of Vienna to study science and math. 2 years after going to the University of Vienna, he returned to the monastery, where he also became a high school teacher. So if you can imagine having Gregor Mendel, the father of genetics, as your high school science teacher. That must have been real special for those people way back in 1853. But also, not only was he a high school teacher, but he worked in the monastery in the garden, and he noticed a few things about the pea plants that he was studying. And he came up with some very good scientific observations, and through his observations, he developed the theory of heredity. And so we're going to go step by step through what Gregor Mendel discovered. Okay? First, he noticed that plants did have different physical characteristics, and he called these physical characteristics traits. He noticed that some plants maybe were tall, some plants were short. He noticed that some plants had purple flowers, some plants had yellow flowers, and he became very interested in where these traits came from and how they would combine to make baby plants. So the first thing he decided was if he was going to study these traits, he wanted to make sure he wanted to know what traits were present in the plants. So he created what was called a purebred organism. He created purebred plants that would produce the same trait. The definition of purebred is an organism that always produces offspring with the same form of a trait as the parent. So for Gregor Mendel, for example, if he was looking at green plants, he would say that if I was going to mate a green plant and a green plant together, then I would always get green plants. So a purebred organism is an organism that has the same trait. In this instance, we're looking at the color green because there were other colors in the plants. But he found out that if he kept breeding the green plants with the green plants, self-pollinating them, using the pollen from the same plant to fertilize the same plant that it came from. So by self-pollination, he got some purebred green plants. Green plants plus green plants always equal green plants. So that is the definition of what a purebred organism is. Okay? He also did the same thing for his yellow plants. And I guess you could imagine that he found out that a yellow plant, when breeding with a yellow plant, always produced a yellow plant. So yellow plus yellow always equals yellow, and green plus green always equals green. And that's how he determined what a purebred plant was. And he did this over many generations, not just 1 or 2 times, but over many years, so that he was very positive he had a purebred plant that would only produce either yellow plants or green plants. So that was one of the first things he did. Okay? And from his studies, he reasoned that there are factors that determine your trait. Okay? And he was thinking, okay, there's got to be a factor on the pollen, which is the male part. There's got to be a factor for the female part also. So he determined that there are factors that determine the trait, one coming from the mother, one coming from the father. And scientists today call those factors genes. Okay? And we're not talking about the genes you're wearing. We're talking about the genes in your cells. Okay? Genes, the factors that control traits. Okay, remember we're talking about traits in plants. We're talking about possibly whether they're tall or short, whether they're yellow or green, whether they have purple flowers, yellow flowers, all these different kinds of traits. Mendel was very concerned with them only. The physical appearance of a trait he called the phenotype. So if a plant appeared to be purple, the phenotype of that plant would then be called purple. Because phenotype simply is the physical appearance or visible trait. Okay? The visible trait in these plants could be yellow. So the phenotype of this plant is yellow. The phenotype of this plant would be green. So when we're talking about phenotype, we're talking about the physical appearance. It could be the color of your hair, the color of your eyes, how tall the plant was. There are lots of different characteristics. But remember, if you put the pH together, the physical appearance with phenotype, it's the way something looks. So that's a good way to remember that phenotype talks about the physical appearance, the way a plant will look, or an animal, or any other trait. Any trait that appears is the phenotype. Okay, but now he's noticed that he's got some green plants and some yellow plants. So there must be a factor that is for yellow plants and a factor that was for green plants. So he came up with the idea that there may be two different factors, one from the mother and one from the father. And they're both for the same trait. They're both for the color of the plant. So instead of calling them both genes, he called them alleles. Alleles are the different form of a gene. In other words, it could be the factor that makes the plant yellow or the factor that makes the plant green. Each plant has a gene that determines its color. And the allele is what determines which color it is. So there would be a white, a yellow allele and a green allele. Guess we're talking about the same trait but a different gene in the cells. Okay? And so he did a few crosses, and I'm going to go through a purebred cross and show you what he determined and how he determined that the plant was purebred. And I want to review a little bit what I did last week. On the board here I have what's called a Punnett square. It is a square that determines the probability, the possible outcome of what happens when a mother and a father mate to produce their children. Now if you remember, last year we talked about what a gamete, or last week, we talked about what a gamete was. A gamete is a haploid cell, also called the sex cell. A haploid cell, also called the sex cell. This would be a parent plant. When the parent plant produces his sex cells, remember they become haploid, they split up. So this parent plant, which is purebred for green, represented by a big G, can only produce sex cells that have a big G. So this plant could produce this sex cell, and this plant could produce this sex cell. And these represent the sex cells of the plant. And a lot of students get really confused out here when they do this. They think this is the father, this is the mother. But no, this is the father, and these are the father's gametes, or the sex cells. So do not get confused with that. And I have the same operation on this side. I have a double dominant mother, purebred green, that can only produce green plants. So in order to make a cross, what we do is we'll take one of the sex cells from the mother, and we will take one of the sex cells from the father, and we will combine them together. We will combine them together to form one new individual. And this new individual then, oops, if I get the pin through the hole there we go. This new individual now has a sex cell from the mother, a sex cell from the father, and the phenotype, the color that appears, is green. Okay? And we can do this 4 times for a purebred dominant plant. If we do this 4 times, we should end up with And if we do this cross correctly, we end up with 4 plants, all identical, all purebred. And this is the idea of what purebred means, that every time a green plant mates with a green plant, the probability of getting a green plant is 100%. So we could go through and talk about what genotype and phenotype means. We know that phenotype, as we talked about earlier, phenotype is the physical appearance. So when I went through these squares, I could do a phenotypic ratio and say I have 1, 2, 3, 4 green plants and 0 yellow plants. So my ratio is 4 to 0. And on a purebred crossing like this, you will always get 100% purebred, and that's the definition of what purebred is. And I could do the same thing for yellow plants, and would you be surprised if I got the same thing? I would end up with 4 yellow plants and a genotypic ratio of 4 to 0. Well, Mendel did this, and when he did this, he called these first generation the P1 generation, or the parental generation. And when he crossed a purebred dominant with a purebred recessive, he got some interesting results, and that's what we're going to look at next. So I'm going to remove my recessive, or one of the parent's genes, and we're going to replace this with a hybrid, or with a yellow plant. So we know that the parent starts off with two alleles for the characteristic and produces babies that each have, I'm losing my, okay, produces babies that only have the recessive G, or the, what we call the recessive trait. And actually I need to talk about that a little bit, because what's going to happen here will explain the difference between a dominant allele and a recessive allele. Well, what happens when I combine the greens and the yellows? What do you think is going to happen? Okay, well, I have a green gene and a yellow gene, and we're going to combine them together. And what do we end up with? We end up with a green plant. And I have the wrong green. That'll work anyway. We end up with a green plant. If the picture's lined up perfectly, we would have a green plant. There we go. This one's the right one. The green masks the yellow. So when the genes are combined, they have the recessive gene and the dominant gene, but what you get for a phenotype is green, because green is dominant, is stronger. So when we do this cross, we end up with we end up with 4 plants that are green, but they are carrying both recessive genes. So the F1 generation, he crossed green with yellow, and the phenotype for all these plants were green. And it left him wondering, how can that be? How is that possible? You're getting 1 factor from the mother, yellow, 1 factor from the father. You would expect maybe some plants to be yellow, some plants to be green, but that didn't happen. So he came up with this idea of dominant and recessive genes. A dominant allele is a trait who always shows up. Okay? If I have a dominant and recessive traits together, the dominant gene always shows up. Whereas a recessive allele is a gene who is masked when it is present with a dominant allele. So the recessive allele is an allele whose trait is masked. And one thing I like to do with my students when I talk about these traits is we talk about how the dominant genes are represented by a capital G, and the recessive genes are represented by a little g. So when we talk about dominant, we talk about dominant being big and strong and overpowering, and it masks or hides the recessive gene. The recessive genes are tiny and small. Recessive, dominant, recessive, dominant. You know, you can get that if you think about it. And why do you think they use letters instead of numbers? Is there a capital, is there an uppercase 1 and a lowercase 1? No, but there is an uppercase in the alphabet and a lowercase. So for the dominant genes, they use capital letters, and for the recessive genes, we use little letters. It's a good way to remember if you think about dominant being strong and overpowering, and we come up with the F1 generation, the first kids that all ended up being green. Okay? Now, what Mendel discovered, and we're not going to go through this entire cross and do another cross, he discovered that when he bred the F1 generation, we have a hybrid cross. In other words, the original parents were green, but they contained the big G and the little g. That means that they would produce sex cells that either had the big G, the dominant allele, or the recessive allele, and the same thing would happen on the top here. We would have a dominant G and a recessive G here. Well, what happens when I combine a big G with a little g? We end up with this cross here. We end up with a green plant that is hybrid or has one of each allele. When we come over here, however, what do you think we end up with? When this allele meets this allele in this box, what is it that we end up with? It's correct. We end up with a yellow plant. A double recessive plant that has both small g's. Okay? Then down here we would end up with a double dominant plant. So we can get rid of this and combine two together to get a double dominant plant. The big G and the big G meet together here. And over here I end up hybrid also. Okay? So he discovered that when he crossed the F1 generation, the plants that were from a yellow mother and a yellow father, that all those plants were green to begin with, but when he crossed those, he ended up with 1 yellow and 3 green. So our phenotypic ratio would then be 3 to 1. But our genotypic ratio, how many double G's do I have? I have 1 double dominant. I have 1 double recessive. And I have 2 hybrid plants. So out of the 4 plants, 1 would be double recessive. So you'd expect 1 out of every 4 plants on that cross to be double recessive. And that's really a pretty good summary of Mendel's experiment. And I like using these overheads and these things because when you combine them, you can really get a good visual of what's going on with the plants. Now, some of you are sitting at home saying, oh, plants are boring. You know, it's nice that we looked at plants and Mendel discovered the laws of heredity by studying plants, but we're more interested in looking at people traits. So I'm going to ask our hostesses to come back up here, and Lewis, and we are going to, if you could, Lewis, could you unclamp that one there for me? Okay, we're going to talk about 4 common traits that are found in human beings. And these traits are expressed either completely dominant or completely recessive. That means either you have it or you don't. And I've kind of checked you out already. Oh,
we have it.
Oh, don't tell my wife that. No. I already checked you guys out. And you have a couple of these traits that I don't have.
Oh.
And Lewis here also is a good example. So the first trait we're going to look at is called the hairline. The dominant trait is a widow's peak. And I happen to have a widow's peak. A widow's peak is when your hairline comes to a point in the middle. That's called a widow's peak, and that's a dominant trait.
No dominant trait.
We're not dominant.
And our twins here, both of them are recessive. Their hairline is straight across.
That's good.
That's not bad. But remember, what we just discovered was that when those yellow plants showed up, the only genotype they could possibly have when it showed up yellow would be double recessive. Okay. So if we were going to do a genotype on your hairline with big H, capital H being dominant, your genotype would be 2 small little h's, recessive. Whereas mine could be dominant, double, or hybrid because I showed the trait. Okay? So all of you can look at your hairlines and your parents' hairlines and see if this trait runs in your family and who carries that trait. Another trait we can look at is called the hitchhiker's thumb. Okay? Thumbs up, everybody. Okay, if we look at our thumbs here, come on closer. Come on, get your thumb in here.
I get the ride.
My thumb is straight up and down, whereas these thumbs are very exaggerated, pointed back towards themselves. Okay, the dominant trait is the hitchhiker's thumb, and the recessive trait is the straight thumb. So if you're going to do a genotype for me with a recessive straight thumb, I would have a double small t, where both of you would either be two big T's or one of each. Correct. Right. So, do you have a hitchhiker's thumb or not? Check it out. Okay, now, next trait we're going to look at is called the earlobes. Okay, earlobes come two different ways. Earlobes come free, which means you have an earlobe that hangs down a little bit. It's not attached. It's just free. And our twins here both have beautiful hanging earlobes.
Just because I wore earrings, you know.
Well, yeah, they could stretch them out a little bit further. You're right. There are factors that does affect that. However, Lewis here is a perfect example of the double recessive trait of having a straight earlobe. His earlobe does not hang down. It is straight across. So Lewis would be double recessive, small e's for the straight earlobe. And mine is dominant, it hangs loose. Okay, the last trait we're going to look at is the tongue trait. The dominant trait for your tongue is rolling your tongue. And rolling your tongue means rolling your tongue.
Making a taco.
Make a taco out of your tongue. Ladies, can you make a taco? Okay, these ladies can both roll their tongue, the dominant trait. Lewis, however, is recessive on his tongue. He can't make his tongue roll.
No talent.
Try. Let's see.
Weak tongue.
And,
no talent.
And you'll notice sometimes in class when I do this with students, there'll be students, you know, some students will just go, no problem. Other students, push it together. The dominant trait, again, is rolling, and the recessive trait would be double R for Lewis. So
we're dominant. And he's weak.
Only for certain traits.
Yeah, we didn't have all dominant.
Only for certain traits.
That's very interesting.
It is. It is very interesting. And it's a fascinating study. You can do a pedigree, go through your family, trace these traits back through your family, and get an idea of where the traits came from.
Yeah. Great. Thank you very
much.
Thank you so much.
My pleasure.
Okay. Oh my gosh, you know what time it is? You know what time it is? Math! Time to solve some problems, I think. Oh, I thought it was time for me to get a watch, but it's time for math, so let's go on over. Greg Gross, how are you? You can do the tongue.
I'm quite dominant, thank you.
Talented tongue. How about with math? You're dominant with math as well.
Yeah, that's about as far as it goes.
What are we learning today?
Well, we are going to be doing problem solving, dealing with the mixture problems.